Recording in progress, people. You are live. All right. So, Unit 1, this covers a lot of the summer work, which of course is probably fresh in your brains. So going straight to our basic chemistry, we've got ionic bonding, covalent bonding, and hydrogen bonding. Ionic bonding, this is where we are taking an electron from a metal, giving it to a non-metal. And because of that, the metal now has more protons than electrons, so it is positive. The non-metal has more electrons than protons, so it is negative. So we now love each other due to electrostatic force, and we form an ionic bond. Covalent bonds, on the other hand, these are going to be where we have typically two non-metals, and we are going to share electrons in the outer layers to form a shared pair. That is then going to hold those atoms together. Covalent bonds are the ones that make up the majority of our biological molecules. So our carbs, our proteins, our nucleic acids, the lipids, they're all covalent bonding. In our proteins, there are some other bonds. There are some ionic bonds and some hydrogen bonds, but the majority of the strong bonds are going to be covalent. Hydrogen bonding, this is going to occur when you have polar molecules. So we see that when we've got water having a slightly negative oxygen and a slightly positive hydrogen, they are going to be attracted to each other, making those hydrogen bonds. This can happen between any kind of polar molecule, and the slightly negative, slightly positive, that is due to unequally shared electrons. So it's still a covalent bond, but we're not playing nice. So the oxygen hogs the electrons, so it becomes negative. The hydrogens become positive, and then the positives of one molecule will be attracted to the negatives of another. We see that in water. That's what gives water all of its different properties. And so when we look at that, we also need to have this molecule being asymmetric. So we've got the negative on one side, the positive on the other side, so that we can form those different poles. That's why it's called a polar molecule. And that's what allows them to be attracted to other things. Our basic properties of water: cohesion, which is water sticking to itself. So it is these hydrogen bonds between the molecules. That's what gives surface tension on the top of water. That's what creates the big drop of water on the penny. That's why we have water drops in the first place, because the water molecules stick to each other, forming things like raindrops and drops from a dropper. In and of itself, cohesion isn't super important for life, but it does work hand in hand with adhesion, which is water sticking to other things. So adhesion, so we see here that we've got a spider web, which is made up of protein. And because proteins have a lot of polar amino acids, the water is going to be attracted to those polar amino acids, creating those water drops. This is a microscopic view of the inside of plant xylem. And so all of these tubes, these are tubes of cellulose. Remembering that cellulose is made of glucose, which is a polar molecule. So water is going to want to stick to the inside of these tubes. And then together, the adhesion and the cohesion are going to cause transpiration. So we've got xylem on the inside of the veins of a leaf. Water can flow out of that xylem to fill the airspace inside the leaf. And we need to have water in there because it's going to keep the leaf moisturized while we have CO2 and oxygen going in and out of the leaf through the stomata during transpiration. Then that gas exchange, again, is what's necessary for the CO2 to come in for photosynthesis, then the oxygen to exit so that we can do things like breathe. That process of getting the water up, so starting in the roots, the water is going to flow into the roots. That is, yes. We are on, so we are in question, so this is question 2. And then we see that I've now jumped to in-depth question number 1. So that's going to be on the back. I did not tell you that. My bad. So there are in-depth questions.
So we jumped.
We did. I didn't tell you that we had these up here.
Can you read this part? Can you start over from? Wait, finish.
We jump from adhesion to in-depth question number 1, which is in the back.
On the back page, the second to last page.
Right.
Yeah.
So our in-depth, so this is explaining transpiration. So we see that water is flowing into the roots. Water is flowing in because there is sugar and salts inside the roots. So that is a lower water potential. So the water flows in, the water flows into the xylem, and then all of our water molecules, we can see there's this big long line of water molecules. They are all connected by cohesion. They are also stuck to the side of the xylem wall by adhesion, so the water does not slide down the inside of the xylem tube. Up in the leaf, as the water is exiting the leaf, the water molecules pull on the water molecules behind it. So if I grab this line and I pull it, it pulls all of these up. And as we were pulling all of these up, that draws water into the xylem and the roots, and it continues that flow. The pulling up is transpiration due to cohesion and adhesion. And we need this to happen to get the water into the leaves so we can have photosynthesis, which is the basis of our primary productivity for our ecosystem. So if we don't have cohesion and adhesion, we don't have transpiration, we don't have photosynthesis, the ecosystem crashes. So they may seem like tiny properties of water, but together they're very important for this whole.
How does the polarity of water factor into that?
Polarity is what makes them cohesive and adhesive. So that's what allows them to pull up and then stay stuck to the side of the xylem. All right. Back to the properties of water, and also in-depth question number 2. Ice floats because water is less dense as a solid. As water freezes, the water molecules organize and they form a crystalline lattice structure. And as these hydrogen bonds between the molecules become more rigid, that forces the water molecules apart. Because the water molecules are forced apart, they take up more space, but they have the same mass, so greater volume. If I increase my volume, but I maintain the mass, then my density is going to go down. So therefore, ice is going to float. Why is that important? This is important for aquatic biomes because that keeps the lakes and the ponds from freezing solid, keeps the oceans from freezing solid. And so you have aquatic liquid water underneath so that the organisms can stay alive. Otherwise, you would have a lot of dead fish. Are we ready?
No.
Are we?
No, what goes on the question 2 and the IDQ 2? Wait, what is the high specific heat? Did we talk about that?
I have not talked about that yet.
So what was the polarity of water equal to?
The
Earlier
polarity of water, what do you mean equal to?
I thought it's so. Yes.
All right. In-depth question number 2 is about turnover. Turnover is what happens in lakes. So during the spring and during the fall, the water temperature is the same, so the water can mix. When the water is mixing, it is turning over. So this is turnover. And this is turnover. Now, during the winter, the coldest water, which is 4 degrees Celsius, sinks to the bottom of the lake. The coldest is going to freeze at the top. So remember, that was different. Densest versus coldest. Densest is 4 degrees, still liquid. Sinks to the bottom, coldest freezes, and we get this thermal stratification. Thermal stratification simply means you've got different layers that are different temperatures. Winter goes on, and then as the water thaws and warms up, it's all 4 degrees, and now it can evenly mix. This is important because what's going to happen, oxygen is going to come down to the bottom, and then decomposed nutrients are going to go to the top. This brings nutrients to the top of the lake for primary productivity to drive the algae, and then the oxygen goes down for the decomposers to continue the process of decomposition. In the lakes, we get stratification again, warm at the top, we get lots of primary productivity, stuff is growing, stuff is dying, dead stuff goes to the bottom, starts to decompose, and then again in the autumn, when the water is the same temperature, it turns over. So the reason that this is important, it continuously brings the turnover, brings oxygen to the bottom for the decomposers, and brings nutrients to the top for primary productivity. Yes.
Where does the colder water go?
At which season?
Or
So it depends on the temperature. So 4 degrees is when water is densest. So it's going to sink. But then when everything is 4 degrees, it can mix evenly. All right. Properties of water due to specific heat. So remembering that specific heat is how much energy it takes to heat something up. Water has a high specific heat of 4.184 joules per gram degrees Celsius. You're not going to need to know that. But this is why water takes longer to heat up than metal. Metals have low specific heats. This is important because it helps regulate the Earth's temperature and it helps with evaporative cooling. As the ocean waters warm up, it causes them to cycle. This creates the ocean currents. That's going to bring warm water to cooler regions, colder water to warmer regions, and then that is also going to help do turnover in the ocean to bring nutrients to the surface. Evaporative cooling is when water evaporates. It has a high heat of vaporization, which means it takes a lot of energy for it to evaporate. So when that sweat is evaporating from your skin, it is removing a lot of heat from your body. So this is going to be important for homeostasis. When leaves and plants are transpiring, that is also helping to cool down the leaf and the plant. And this is why when plants are stressed and they close their stomata because they don't have enough water, this is why it can lead to damage to the plant because the plant can't sweat. It can't cool down, and that heat or that leaf can overheat and start to denature the enzymes in the leaf. It's just like if you weren't able to sweat, you would overheat and you would die. With our ocean currents, what we see is we have warm ocean currents coming up here. We get up to the Arctic, they cool down, and then the ocean currents drop down to the bottom of the ocean. We get the cycling as the water cycles down at the bottom of the ocean. It's picking up nutrients, and then it begins to rise. And right here, as that water comes up, it brings all those nutrients to the surface, and that drives primary productivity. As that drives the primary productivity, huge amount of plankton growth, huge amount of fish growth. This is why whales migrate off to the coast of Alaska. That's why there's great whale watching, because there's so much productivity going on there because of this turnover. If the ocean currents were to shut down, the ocean productivity would drop tremendously. And that is bad because this is where 50% of the oxygen of the planet comes from. So if the oceans get too warm, that could potentially shut down the ocean currents. Yes. Mm-hmm. So they will go up there, get really nice and fat, and then they come down and then they reproduce where the water is warmer. All right. The same thing with on the coastal regions. So the ocean air is cooler because the heat from the land causes the air to rise, then it gets up in the atmosphere and it drops. And this creates these cycling wind currents, which is why it's almost always windy at the beach, because you've got these currents. It helps moderate the temperature along the coast, which gives you milder climates versus, in the Midwest of the United States, super hot in the summer, super cold in the winter, because you don't have these large bodies of water to absorb and release heat and help regulate the temperature. Water is an excellent solvent. The polarity of water attracts it to other charged things. And this is extremely important inside the cells because as the water dissolves components, it makes them more readily available for chemical reactions. Dissolving things is basically increasing their surface area so that they can react faster. Things that like water are hydrophilic, so that would be ionic things, polar things. Things that do not like it are hydrophobic or water-fearing. This would be anything that doesn't have a charge or non-polar substances. There are some things that are hydrophilic that don't dissolve, like cellulose, because the molecules are just too big for the water molecules to surround and separate. Cellulose fibers are extremely long, but they do bind to water, which is why cotton towels work, paper towels work, unless the ones that we have in the lab and they just smear the water around. But hydrophilic, hydrophobic, this is going to be extremely important when we get to cellular transport and the membrane structure and our phospholipid tails, et cetera. Why do we coat things with wax? Because wax is hydrophobic and it waterproofs it. Correct. Think about skateboards, surfboards, something. Did you say because it waterproofs them?
I'm saying,
No. All right. So what we see here, we've got an ionic compound. This is salt, sodium ions, chlorine ions, and we see the negative oxygens are surrounding the positive sodium. The positive hydrogens surround the chlorine, which is negative, and that separates them. That is the process of solvation, and that makes those readily available for cells to use, particularly in-depth question number 6. So So in our nervous system, we need sodium and potassium, and dissolving these compounds to make the ions. Ions are what give the charge differential across the nerve membrane. So when we look at this, the role of water as a universal solvent, cellular transport, and an action potential. So remember, action potential is your nerve. So in order for this to work, we need to have ions. And these create the charge differential. As the sodium potassium pump, we are pumping 3 potassiums or 3 sodiums out for every 2 potassiums in. We get a difference in charge. That's what's meant by charge differential. That is what makes our nervous system work. So the active transport pumps those ions across. We get our 3 sodiums to 2 potassium, 3 sodium to 2 potassium ratio, and that's a difference in charge. And then when the nerve signal fires, we get the flip-flop of the sodium and the potassium. That's what's going to transfer the signal down the nerve cell to the end to give us to tell our brain something. But if these ions weren't dissolved, if sodium and chlorine were together, what charge would they have? They would be neutral. And if we have neutral charges, can we get a charge differential? No. So they have to be dissolved as ions in order for those cells to work. And why do we have to do active transport for this? 2 reasons. We need the gradient, so we have to build those gradients. And then what's the other reason? That's what makes it active. We're using ATP. Why can't the ions just go across? What about
the tails?
So the tails are what? Hydrophobic, and ions are hydrophilic. Can ions cross the tails? No, so they have to be pumped across. All right. Some large things like proteins, they can be surrounded by water and they can then be dissolved into liquids. So the protein that is in milk, those are soluble proteins. They are huge molecules, but they are polar, so they get surrounded by water and they can be dissolved in the water. Because proteins have different kinds of amino acids. So we have non-polar amino acids. We also have polar amino acids. And we have electrically charged amino acids. So if a protein is soluble in water, if we go back to this picture, where would the non-polar amino acids be? On the outside of the protein or on the inside of the protein? On the inside, because the charged proteins have to be on the outside. These are charged proteins. And then the non-charged ones have to be on the inside, away from the water. And so what happens with milk proteins also have these electrically charged amino acids on their surface. As bacteria begin to break down the sugars in the milk, they create acids. They create hydrogen ions, which begin to bind to these charged amino acids, making them neutral. And now that they are neutral, they no longer dissolve in the water and the proteins begin to clump together. The reason milk spoils and gets clumpy is because the pH has shifted, making the proteins non-soluble anymore because they have changed the charge. Speaking of acids and bases, jumping to in-depth question number 3. Acids are either going to give off hydrogen ions or they absorb hydroxide ions. Bases either give off hydroxide ions or they absorb hydrogen ions. And what happens when you combine an acid and a base? They do not explode. They neutralize each other. So the pH scale is the perhydrian scale. That's what pH stands for. And it is the concentration of the hydronium ion. Hydronium ions are formed when you have a hydrogen plus a water molecule, and that forms H3O. The more of this you have, the more acidic it is, because pH is calculated using the negative log of the hydronium ion concentration. Theoretically, you could be tested on that. It has never shown up on the AP test before. But on the equation sheet still, maybe it's not on there anymore. There it is, negative log of hydrogen ions. So if they told you that HCl, if it is a 3 molar solution, then you would take the negative log of 3, which would be actually very acidic. If you had pH, if you had a HCl concentration of 0.01, that would give you a pH of 2. So this would be the concentration of your stomach acid. And so if you did the negative log of that, that would be a pH of 2. So the higher this concentration, so if you did 3, that would be even higher. You get a negative pH, which is possible. And then bases, if you have a pH of 2, the pOH, which is the scale of base, would be a 12. We don't need to know that. Why are acids and bases important? Did you say homeostasis? It is. So specifically, stomach acid that kills stuff, ocean acidification, what? Enzymes, most importantly, yes. Our pH scale, if we go from a pH of 5 to a pH of 6, pH of 5 is 10 times more than 6 because log is a 10 scale, or is a 10 power of 10 scale. So a pH of 4 is 10 times more acidic than 5, which is 10 times more acidic than 6. So 4 is 100 times more acidic than 6. Our pH scale, so we've got acid is below 7, bases are above 7, human blood is about 7.35. That's very tightly regulated because of our enzymes. So our kidneys help regulate that, our breathing helps regulate that. And so our typical biological range is 6 to 8. Above or below that is death, unless you are something like an acidophilic bacterium, which really likes acidic environments. Buffers are things that regulate pH. AP Chem people, that's all you need to know about them. They absorb or release hydrogen ions just to keep it in check. The thing in our body that is the main buffer are the amino acids in our blood, because they can give off hydrogens or they can absorb hydrogens. and they help regulate pH. Yes.
Why?
Rainwater, that's a good question. That ties into one of our in-depth questions. Rainwater is naturally acidic because um, we'll get to that. We'll come back to that when we do ocean acidification. So we've got polar and electrically charged. So these are going to be the ones that act as buffers in our blood. This is also important because this affects the transport of some molecules. So just really quickly, when you have a carboxylic acid group, which is a carbon double bonded oxygen, and here it has a negative. If you get a more acidic environment, this is going to gain a hydrogen and become neutral. This molecule can then bypass through cell membranes. If you have an amino group in pH, low pH, it becomes positive and won't go through the membrane. If you put them in a basic group, this loses the hydrogen, becomes negative and won't go across the membrane. This becomes neutral and will. So these are functional groups, amino group, carboxylic acid groups, and pharmacists put these onto medications to help determine where they are absorbed in the body. So if they need to be absorbed in the stomach, a pH of 2, we give them a carboxylic acid group so they'll become neutral and move across the stomach lining. If they need to be absorbed in the intestine, you put on an amino group, which in a basic condition will become neutral and cross the intestinal membranes. So those functional groups tied in with pH help with medical deliveries. pH and enzyme function. All enzymes have an optimal pH. What happens if you are outside of the optimal pH? We are in that question number 3 still. It's going to denature. And what do we mean by denature? What's that mean for change the shape, change the function. So it destroys the ability of the enzyme to catalyze reactions. So pH of pepsin in the stomach is the optimal pH is 2. Trypsin, which is in your large intestine or your small intestine, that is going to be a higher pH of 8. Ocean acidification, this is when we have carbon dioxide and water form carbonic acid. So this goes to your rainwater question. So in the atmosphere, carbon dioxide mixes with the water in the atmosphere, it becomes carbonic acid and is slightly acidic. So that's normal. When we talk about acid rain as an environmental problem, that is where excess fossil fuels, in addition to CO2, gives you nitrogen compounds and sulfur compounds, which with water, actually this is SO3, with water gives you H2SO4, which is sulfuric acid, and then with water, that should probably be a 2, and that gives you nitric acid. And so fossil fuel pollution, nitrates and sulfates, that gives you more acidic rain. Natural rain should have a pH of about 5.6, and that's from the carbon dioxide. But when we start pumping more CO2 and more of these in, that drives down the pH of the ocean, and then that messes with the availability of carbonate, which is necessary for building corals, clam shells, mussel shells, and then the ocean becomes too acidic for things to live in. When we look, okay, question 3. So much time. So hydrogen bonding is also important for our protein structure. We've got our primary structure, which is our sequence of amino acids, but we also have our secondary structure, which is the hydrogen bonding between the amino acids that form the pleated sheets in the spiral. So if you remember back to that paper thing we did, where we cut it out, we folded it, twirled it, put them all together. That gives us our secondary structure of our protein. And then our secondary structures build to our tertiary structures and our quaternaries, which gives us our final shape and our final function. In DNA, this holds our thymines and adenines together, our cytosines and guanines together. There's 2 between A's and T's, 3 between G's and C's, and this is what holds the 2 sides of the double helix together. All right, our biological molecules. So our biological elements are the sponge elements. Again, remember these are all held together by covalent bonds. When we are building biological molecules, we are going to use the process of dehydration synthesis. So we've got a hydrogen and a hydroxyl, or maybe this is another hydroxyl. We are going to remove a water, and that forms a bond between them. We are removing water, that's dehydrating. We are building a larger molecule, that's synthesis. The opposite of this is when we put the water back in, and that breaks the bond. So breaking in biology is lysis. And we are using water to do that. So hydrolysis, hydrolysis, is splitting with water. And this requires enzymes. This is digestion. So when your body is breaking down starch, pulling the glucose molecules apart, it's adding water to them and separating the individual glucose units off of the starch so you can absorb them. Yes, all of these are on my Baylor as well.
What is dehydration synthesis?
So that's to build molecules. This is how we build starch. This is how we build fats. It's how we build proteins. It's how we build DNA. It's all dehydration synthesis. Hydrolysis is how we break everything apart. We put the water back in between the 2 molecules. All right, carbohydrates. This is what you're writing down right now. Energy, storage, and structure. Those are our main things. All of this other stuff, you're going to have to go back and write in. You're going to have to go to the PowerPoint online and fill that in. So we use energy for cell respiration. Storage, we are storing glycogen. If we're animals, we're storing starch. If we're plants, structure, we're talking cellulose for plant cell walls, chitin for fungal cell walls. Lipids, energy storage, so fats and oils. We also have fats for structure as the phospholipids for our cell membrane, and then the chemical hormone function of lipids is the steroids. And that's going to be part of our signal transduction pathway, which is not in this unit. The functions of proteins. All you're doing right now is writing functions. You can write examples down later. Remembering between saturated and unsaturated fats. Unsaturated fats have that double bond, which changes the structure so they can't stack and they stay as liquids. So our unsaturated fats are oils, saturated fats are fats. Phospholipids, these are going back. Question 12, difference between saturated and unsaturated fats is the double bond. which changes the angle of the tail so it can't stack. Unsaturated is a double bond, yes. Phospholipids, extremely important because they are amphipathic. They are going to be hydrophilic and hydrophobic. So the hydrophilic heads, hydrophobic tails, that creates our phospholipid bilayer, which separates the inside of the cell from the outside of the cell. That's what makes life. If we did not have phospholipids, we would not have cells. Doesn't matter if you had DNA, doesn't matter if you had enzymes, doesn't matter if you had the, whatever. If you don't have a separation from the external environment, you can't have the components of life contained. Proteins, structure, storage, transport, structure, all your cell parts. Majority of them are made of protein. Storage, we store amino acids in our muscles. Transport, we're talking transport proteins for active transport, passive transport, talking movement of exocytosis, endocytosis, also just walking, that's transport as well. Communication, hormones in the signal transduction pathway, our peptide hormones, movement with cilia and flagella on cells. Also, we have protection in the form of antibodies in our immune system. And most importantly is their role as a catalyst and in metabolism with enzymes. Question 7, our basic structure of the amino acids. We are looking at the carboxyl group. The amino group. And then this, depending on what it is, this is going to be the R group, which is what's different from one amino acid to the next. It's not always that. It could be just a CH3. It could be an S. It could be something completely different. The carboxyl group, this is acidic. So that's why it's an amino acid. And this is going to allow us to form proteins by dehydration synthesis, and that forms the peptide bond right there. As we build our peptide bonds, we are going to get the primary structure of the protein. That's the sequence. So when we did the central dogma, and we talked about translation and the putting together of the sequence of amino acids using the ribosome and the A site and the P site and the E site and the anticodon and the codon, that's building this primary structure. This is just our sequence of amino acids. Those amino acids start to interact with hydrogen bonding to form our secondary structures, the pleated sheets and the alpha helixes. Those begin to bond together in the tertiary structure, and this is due to interactions of the R group. For some proteins, that's it. We hit a tertiary structure, we have our shape, we do our function. Some of them bind together in clumps, and that's our quaternary structure. And that gives them their specific particular structure and function. So we see here in depth question number 4, we have different bondings. So these are the R groups between the amino acids. We've got an ionic bond right there, bonding those together. We've got a hydrophobic interaction there. 2 hydrophobic groups like each other because they are trying to get away from water. And then we've got hydrogen bonding, and then this disulfide linkage that is covalent. Out of these 4, hydrophobic, polar, ionic, and covalent, which is the strongest bond? It is covalent. That is your strongest bond. What is your weakest bond in all of this? It's hydrophobic. There's nothing holding them together. There's no charges. There's at least some charge here and a stronger charge here. So this would be our weakest, second weakest, second strongest, and our strongest. This is what gives shape to the proteins. This is what gives it function. What does denaturing do to this whole setup? Changes that bonding pattern, which then changes the shape and changes the function. So how does it relate to proteins and transport? Tertiary structures, those are our transmembranal proteins. That's what determines what can go in and out. The shape of the protein that determines what is transported. So if we change what's going on in there, and this is what happens in cystic fibrosis, there is a mutation that changes these pores. Therefore, cystic fibrosis cannot transport chlorine out of the cell into the mucus. That chlorine is supposed to draw in water to make the mucus thin. But because that does not happen in cystic fibrosis, you get this thick, thick mucus building up in the lungs, which causes all the problems with cystic fibrosis. All right, we're not done. That was the 2-minute warning. DNA, hereditary molecule. RNA regulates everything by regulating protein synthesis.
What is this?
This is question number 6.
No, like, what do we label on top? There's like, probably the proteins.
You have 3 blanks for that one? Oh, that's just cross that out. So,
This is like DNA.
this is nucleic acids. Yeah. And so that's gonna, kind of run the show. It's kind of important. I will finish the rest of it, and then I will post the video. You can watch it, and then there are going to be some relationship questions that you might have to finish on your own. Pause for a moment. All right, moving on. So our basic nucleotide structure, we have phosphate group. We've got some type of sugar. This could be deoxyribose for DNA or simply ribose for RNA. And these two are going to make up the sides of the DNA or the RNA. And then you have your nitrogenous base, which could be your purines, which are your adenine and your guanine, or your pyrimidines, which could be your thymine, cytosine, or potentially uracil if it is RNA. When we look at our basic system, we see our deoxyribose and our phosphates over here and over here. And then we've got our pairing of our GUCs, A's, and T's. And then, of course, the big important thing is going to be the 5' and the 3', giving us the directionality based on the carbons from the sugar. And of course, everything is built 5' to 3'. When we are copying DNA to DNA, we copy in the 5' to 3' direction. So the DNA, the original DNA is read 3 to 5, while the new is built 5 to 3. When we are transcribing RNA, the DNA, this is 3, 5. So we are building 5 to 3. And when it feeds through the ribosome in translation. The 5' end goes in first, then the 3' end goes in at the end. So we feed through that way. So all of this directionality, this is what allows our polymerases, DNA polymerase or RNA polymerase, to build the molecules in a unidirectional way. And of course, that is going to cause issues when we are replicating DNA and we are going the opposite way and we get those lagging strands and the Okazaki fragments. When we are copying our DNA, we are going to have what is called the coding strand and the template strand. The template strand, this is what is copied into RNA. So the 3' to 5' side is copied 5 to 3. So that gives us the RNA. And then when our tRNAs come in, if we back up here, so this is 5 to 3. So this is 5 to 3 matching on the anticodon. We are going to talk about different names for coding and template strands later. Scientists have named them 3,000 different ways, and it's confusing, but we need to go over those later. DNA is double-stranded. RNA typically is single-stranded. We have deoxyribose in our DNA. We've got ribose in our RNA. The ribose is different because it has an OH on the second carbon. We only have a hydrogen on the deoxyribose. That oxygen has been removed. Deoxyribose. Common nucleotide bases are C, G, and A. RNA has uracil, thymine for DNA. Basic relationship questions. How are the subcomponents of the biological molecules related to the movement and cycling of nutrients in the ecosystem? So we've got our sponge elements. So sulfur and phosphorus are going to be in the sulfur and phosphorus cycles. So this is where we have rock. It is going to erode, and that gives us sulfate and phosphate in the soil. Plants absorb those, incorporate them into proteins and into nucleic acids. Oxygen, this is going to come from the water cycle with our hydrogen absorbed by plants. Photosynthesis, that makes it available to be built into other things. Nitrogen from the nitrogen cycle. And so N2 is going to be fixed into NO2 minus and into NO3 minus. This is nitrogen fixation. This is done by bacteria. The nitrate then goes into plants and it cycles through, and the plants make it into amino acids and nitrogenous bases, and then that cycles through your food chain and your food web. Carbon, CO2, so water and CO2 through photosynthesis and cell respiration makes up the carbon cycle and gives you the basis for everything. How do humans screw things up? So many ways. Sulfur and phosphorus, burning fossil fuels, pumps sulfur into the air. Phosphates from fertilizers, fuels, fertilizer. Nitrogen fixation is also fertilizers. Excess fertilizer, that's going to lead to eutrophication and algal blooms. Dead fish. And then excess CO2, climate change, global warming, hurricanes, death, fires, terror, etc. Humans. All right, systems become more complex, new properties that were not in the simpler become apparent. So these are emergent properties. So structure folding of a protein. So again, we've got that primary structure, just the sequence of amino acids. As this folds from primary to secondary to tertiary to quaternary, we get new shapes. New shapes mean new functions. This primary structure, that is from DNA to RNA to protein. That is our central dogma. So if we change our DNA, give a little mutation, we can change our RNA, we can change our primary structure, our secondary, our tertiary, our quaternary, and then we can get a new shape and a new function. And that could be something like our good friend, sickle cell anemia. Or maybe something positive, and you get a new mutation, which then gives you some new raw material for natural selection. Phospholipid, significant for life on Earth. Phospholipids make up our cell membrane. This is the cell membrane. So once we get our cell membrane, then we can get stuff inside that is different from stuff outside. RNA, proteins, DNA, then you get simple metabolism. Then you get your first simple cell and then natural selection, and you get Mr. Irvin. Hooray. Thank you, phospholipids. All right, so how does the molecular charge of lipids and proteins relate to processes of the signal transduction pathway? So again, we've got two types of signals. We've got protein hormones or amine hormones, and we've got steroids. These are lipids. These have a charge. These are not. These will bind to the surface. And they will initiate the signal transduction pathway. Steroids, they are going to go through the membrane. And they will have an internal receptor to start the signal transduction pathway. So can it go through the membrane or not? It cannot, so it binds to the surface. It can, so it starts the signal transduction pathway inside. All right, good luck.